It's a real pleasure to welcome Roger back to Northwestern once again. Roger went to school here and was my advisor, but a number of us have a relationship with Roger, so it's really wonderful to have him here visiting us again. He's the Cecilia and Ida Green Professor of Biological and Mechanical Engineering at MIT. He was a founding member of the Biological Engineering Department there. Roger's numbers are really pretty spectacular. Roger has an H index of 133. It's sort of a mind-boggling high number, over 200 publications. He's the 2010 recipient of the ASME Listener Medal for Lifetime Achievement, and he's been elected to the Institute of Medicine. I could go through the list of his contributions, but then you wouldn't actually be able to hear the talk because it's a very long list. He's worked in the area of flow in arteries, veins, airways. I've worked in the area with him in eye field physiology and lung physiology. It's a long, long list. He'd generally been working in the area of mechanobiology, more recently going down the sort of smaller length scales, working on how cells respond and looking at the effects of mechanical stimuli. He is really a special person to work with, and as a mentor, he's really a sort of a unique style that at the same time is laid back but very rigorous, which is why I think his groups are so productive. Today, Roger is going to be telling us about the complex in vitro systems to study neurovascular function and health and disease. Welcome, Roger.
Great. Thank you very much, Mark. It's always nice to be back to Northwestern. I get back about every 4 or 5 years, I think, since I graduated in '72, so it's a few years back. But I'm going to tell you just about some recent work that we've been doing. Starting about 10 years ago, starting working on neurological systems. So I'm going to focus on that and include a fair amount of work that's been done, actually some results we just got last week. So hopefully it'll be interesting to all of you. So I'm going to talk about models that we've been developing for neurological disease. I don't think I have to tell anybody here that Alzheimer's is a terrible problem in the U.S. and worldwide, both in terms of its cost to the world, monetarily, individual families, the cost of caring for people with dementia, and the fact that 1 in 3 of us seniors are going to die with dementia, either because of it or with it. And it's not going to be changing anytime fast, at least according to current projections. By 2050, it's the number of cases of Alzheimer's is predicted to roughly double.
Click the OK on the screen
Oh,
so we can see the title.
I had gotten rid of one of those.
You pop up.
Yeah, I hope it doesn't pop up again. There we go. Okay. So it's definitely increasing in incidence, and as a result, pharma companies have really taken an interest in it. I got to get back down here so I can advance my slides. There we go. Okay, and this just gives you some idea of the number of drugs. There are 164 trials going on for various drugs for Alzheimer's, and 127 drugs in the pipeline for Alzheimer's in 2024. So huge activity. And you can imagine that the pharmaceutical companies are looking for a variety of different methods for screening for new Alzheimer's drugs. A couple of which have come on the market now. The one that I think you've probably heard most about is lecanemab, which is an infusion every 2 weeks, and which essentially gets rid of the amyloid plaque in the brains. It isn't known yet to what extent it actually prevents deterioration of cognitive function. But it's a start, and a lot of other drugs affecting different aspects of Alzheimer's are coming online too. So as you can imagine, pharma companies are interested in ways of modeling the brain. Typically, they've used animal studies, animal models, mice predominantly, some 2D in vitro assays, but really very simplistic models for the most part. More recently, for example, cerebral organoids have come onto the scene that can replicate a lot of important functions of the brain and developed a lot of the different cell types that are present, but are still very limited in terms of both their usefulness in terms of drug screening and also the incorporation of these models into pharma. More often, they're looking to these microphysiological systems or complex in vitro systems where you can model certain aspects of neurological function, vascular function, for example, that I'm going to talk about today, and that are actually becoming useful, I think, to the pharma companies. A lot of the work that we do right now, about half our funding comes from large pharma companies. And several of them have already brought in some of the techniques that we use in-house so that they can use it to mostly at this point screening for drugs that get across the blood-brain barrier, but ultimately also for disease models. And the other thing that's promoting this is the fact that the FDA has now realized that they really need to cut back on animal experiments. So, you know, there are the 3 R's. There's the activities that are underway in the government, the recent FDA Modernization Act. All these things are pushing companies and researchers to go more and more toward in vitro models and human-based models and away from animal studies. Animal studies, it's not just for the benefit of the animals, but also they tend not to be very predictive of human outcome. So you really need to have a better model and preferably a human-based model. So Alzheimer's is associated with a variety of different changes in function in the brain. The main things that you hear about are the generation of amyloid beta plaques, but also these tau tangles, tau protein tangles that actually form inside the neurons and can cause neuronal death. In fact, there's still a debate going on in the community. I just heard a talk 2 weeks ago at the University of Illinois, a neurologist from Hopkins, who basically was saying, I don't believe that beta amyloid has any role really in Alzheimer's disease. It's really the tau tangles that are important. So he didn't have much confidence that the new drugs that are being marketed today are going to have much impact. But we'll see. They have evidence, you know, some decline some reduction in the decline of cognitive function. And certainly they can clear out the beta amyloid. So, you know, we hope that that's going to have some positive effect. We focus largely on the vascular effects, including both the blood vasculature and also the lymphatic systems. I'll show you some of those models. But that also astrocyte dysfunction, immune cell failure, and changes in brain mechanics. The brain stiffness changes over time with Alzheimer's. So all these effects, shrinkage of the brain you see in the middle, all these effects occur progressively over time, and then ultimately the patient would die of the disease, you know, typically in maybe 5 or 10 years after onset. I mentioned that our interest has been largely on the vascular side of things. So we started doing vascular modeling about 15 years ago. And we developed these, we were working with a biomaterial that we didn't have much of, and it was very expensive. So we thought we have to design small experimental systems, small experimental platforms that we could inject the gel into. And we wanted to look at how blood vessels would sprout into the gel, just as a baseline experiment. So in the process, we set up these microfluidic systems that I'll show you in just a few minutes. But ultimately, they've led to a lot of work, both in metastatic cancer, but also in neurological and neurovascular disease, one of which is shown here. And this is sort of the progression that you see with Alzheimer's, going from, on the left, a healthy microvascular network in the brain with basement membranes, pericytes around the outside of the endothelium, and astrocyte end feet that communicate with the vasculature. Over time, you get an increase in permeability of the blood-brain barrier. And oftentimes get these microbleeds that show up in MRI imaging after a while. And also there are changes in the expression of some of these important transporter proteins that are mostly found in the brain or highly expressed in the brain, because the brain is such a tight barrier. It uses these transporters to shuttle important molecules back and forth, both into the brain and out of the brain. And then later on, there's really a general breakdown of the vasculature. But the cerebral amyloid angiopathy, which is sort of the second box there, is something that occurs at the early stages of Alzheimer's and is thought maybe to have some sort of a causal effect on the disease. So I said a little bit about the structure. This just shows it in a little more detail. There's the capillary lumen in the center. There's an endothelial barrier around the outside. And on the outside of the endothelium, you tend to have these pericytes that wrap around at least a portion of the vessel, if not all of it. And there's what's not shown very well here, but there's something called the perivascular space or the glymphatic space that separates the sort of the pericyte layer from the astrocyte end feed. The astrocyte end feed actually form this secondary channel that goes around the outside. And fluid can pass through that perivascular space and get cleared from the brain. So there's a lot of interest in understanding these, what's called glymphatic flow that pass along the vasculature into the brain parenchyma and then along the veins on the other side, emptying out into the subdural space, into the subarachnoid space, and then entering into lymphatics, all of which are located in the meninges, which is just below the surface of the brain, so the surface of the skull. Okay, so that's the mechanism by which the amyloid beta gets cleared under normal circumstances. And obviously, if something happens along the way in terms of either changes in the barrier function of the BBB itself or the inability of the lymphatics to clear the amyloid, then that's going to cause an accumulation and then form these plaques. And by the way, if anybody has questions along the way, please don't hesitate to ask. I don't mind at all. So we started working on this problem about 10 years ago. And I'm just going to show briefly some of our first attempts. On the left-hand side, this is some work that we did in the lab in Singapore that I had, where we put astrocytes in one gel region, neurons in another region, and then an endothelial barrier on the side. It was mixed species. We had rat neurons and astrocytes, and then we had human endothelial cells. Pericytes were not included in the model, and the barrier function was not particularly good. Permeabilities were probably 1 to 2 orders of magnitude higher. In other words, the vessels were 1 to 2 orders of magnitude more leaky than you had in vivo. So we tried something else, still sort of a compartmentalized model where we had 3 different compartments, a media channel, then a channel where we would put the neurons, and then a channel where we had an air region that we could allow the neurons to differentiate in the matrigel on the left as we allowed the endothelium to form on the right-hand side on the side of a collagen gel. That worked a little bit better. We got somewhat better permeabilities, although they were still very high. We did see in this model, if we used neurons that were modified to overexpress A beta 40, 42, that we did see some changes in gene expression, in particular in claudin 5, we saw that there was a reduction. Claudin 5 is one of the main junctional proteins in the brain that maintains the barrier function. And you can see we could also measure changes in permeability associated with the disease model as opposed to the wild type model. And we saw a little bit of evidence here too. The green is some A beta 42 or beta 40 that actually accumulated on the side of the endothelium. But that's about as much as we saw, and those experiments had only been carried out about 6 days after the astrocyte cell differentiation occurred. So we looked for other ways of doing this. And at about that time, we were thinking about cell self-assembly and how cells have these emergent properties that if you put cell populations together, cells of different cell types, they can assemble all by themselves into a lot of the structures that you see in vivo. And we've been doing this with vascular networks for a couple of years already, feeding human umbilical vein endothelial cells, mixing them in with fibroblasts, normal human lung fibroblasts, and the vascular networks would form on their own. So we thought, well, could we do the same thing here? The basic principle here is, and I want to distinguish some of what we're currently doing from some of the more engineered approaches that people are using a lot today. This is what I consider to be engineered systems. What you see there is the lung chip model that was published back in 2010 by Don Inger's group. There's also a biological robot there, where basically you provide the structure in the device itself, and then you see the cells out to it. Or in the case of the biobot, you have the biobot structure, and then you get a muscle strip to grow between these two supports. Okay, so very much engineered approach where you're sort of mandating what the cells do. Sort of an intermediate approach is shown here, where what we did, this was a neuromuscular junction that we developed about 8 years ago, where we formed a cluster of motor neurons, put them into one part of the device, and then we had a muscle strip that we put into a separate part of the device. These are all derived from iPS cells or ES cells. And then the neurons, they sense the muscle. There are factors that are being secreted by the muscle that induce the neurites to go toward the muscle, into the muscle, and form neuromuscular junctions. The cells were engineered to be optogenetic, so they express channel-rhodopsin. So we could shine light on the neurons, and we could get the muscle to contract, to demonstrate that the neuromuscular junction was actually functional. And we use this a little bit to look at ALS and the role of a protein called TDP43 that plays an important role. The other extreme is the organoids themselves, where everything is fully emergent. You just take the skeletal cells and you treat it really globally with growth factors or differentiation factors. And they can differentiate into brain, into liver, into kidney, into lung, different organs, or at least some aspect of an organ. So obviously it's not a functional brain here. And in fact, these brain organoids are pretty much empty in the core because they're large enough that they become the product. So you get these void regions. They tend not to be vascularized, although you can induce vascular structures to grow in these. But so far, people haven't been able to connect those internal vasculatures with an external vasculature to get the perfusion to go through. Okay, so the applications are still somewhat limited, although there's a lot of work going on in organoids. What I'm going to talk to you about in terms of our system lies somewhere in between these two. They're somewhat engineered, but also we take advantage. We do use organoids in our system for the neurons, and also very much rely on the emergent properties of the cells to form these vascular structures. So the types of devices we use, they're all sort of variations on the design that you see here, where we have a central gel region shown sort of in pink there, and then that's flanked by different media channels that we have access to. And that enables us to put various cell types into the gel in the center. We can have multiple gel regions, multiple cell types. We have access to the media to the side, so we can set up concentration gradients or pressure gradients, so we can drive flow through the gel. We can grow vascular networks in that gel, and then we can perfuse those vascular networks. These are my disclosures, but also this happens to be the device that AIM Biotech, it's a design that AIM Biotech now markets, and that we use for a lot of our sort of basic experiments. Although the design here is kind of a variation on this, but it's more like what we currently use for the neurovascular system. We've also had a lot of support from industry, and I think that's just an indication of the fact that pharma recognizes that they need to move to these kinds of systems. The animal testing, first of all, is not very predictive of what happens in humans, and secondly, there's a move to get away from the animals. So we've had support from all the companies that you see listed there, and continuing support, especially in our neurovascular model. So with a model like this, certainly we can do various types of imaging. What you see there is an EM image of a vascular wall showing the cell-cell junction. On the right, it's a fluorescent image of a tumor, a glioblastoma tumor up in the corner, and our blood-brain barrier model down in the lower left. We can do various types of biochemical analysis because we have enough media that we can sample, that we can do cytokine arrays or protein analysis and so forth. We can do RNA-seq, although we typically have to combine the cells from as many as 10 devices in order to get enough RNA to do the analysis. It's a little bit problematic. The students who are doing the work on the brain now, they typically seed in one day. They don't do this every day, but they seed about 100 devices, each one individually. There are ways of making that higher throughput, and we have some higher throughput platforms, but right now we're doing it individually. And then we also do a lot of functional assessment. So for example, with the neuromuscular junction, we would look at force contraction because this muscle was on a compliant post. So we could see the post deflection to measure the force generated by the muscle. And also could obviously identify whether the neurons were connecting with the muscle to see whether we could shine light on the neurons and then get the muscle to contract. The other things you see here, this is a measurement of permeability that I'll talk more about later on. And then we could also look at migratory patterns of cancer cells and various other things in these devices. So I mentioned that we started looking at developing these vascular networks. This is sort of one image. It's not typical. This is probably the best image we see, but it's not atypical of what we can generate now. This happens to be a collagen type 4. It's visualizing the basement membrane of the vasculature. And it's a collapsed Z-stack, so you see kind of the three-dimensionality of the system. And I don't have a scale bar here, but the vessels tend to be, they range from about 10 microns up to about 100 microns, with an average maybe about 40 microns. So large for capillaries, probably small for arterioles or venules. And then up in the upper right, you can see a couple of the EM images that we generated looking. Here we've actually identified vesicles being formed on the, on the border. This is on the luminal side, and then there's a tissue side on the other side. So some of these were actually being transported across. So you can actually visualize that in these models, and similar to what you would see in vivo. One of the things we're interested in is we were growing these systems out to about, at this point, about 2 weeks or so. But we noticed that there were some very dramatic mechanical changes in the system. So we wanted to take a look at that. Working with Richard Heinz's group at MIT, we took the systems, like you see on the right, the red are the fibroblasts and the green are the endothelial cells. There in the upper right, you can see one of the fibroblasts sort of wrapping around one of the endothelial capillaries. And the lower right includes a reflectance image of the matrix. And you can see the matrix has been highly modified. The regions where it's very dense, regions where it's non-existent, probably regions where there was once a vessel and then that vessel regressed. But there's a lot of modification that occurs. So what you start with is not what you wind up with, say, 2 weeks into the experiment. And that's not just in terms of the structure, but it's also in terms of the content of that matrix. What are the proteins that make it up? What we did is we used mass spec to analyze a case where, in the case of the co-culture on the far left, that's with the endothelial cells and the fibroblasts. And you can see we got a dramatic change in a lot of these matrix proteins. Whereas if we grew either a monoculture of the HUVEX, the endothelial cells, or the normal human lung fibroblasts, we saw very little effect. So here, clearly the two cell types are they're bidirectional. There's a lot of bidirectional signaling going on. As a result, after 2 weeks, there was about a 33% increase in the matrix. The matrix that we looked at, 33% of it was newly synthesized by the cells, and about 67%, or about 2/3, was the original matrix that we started with. But of that 1/3, you can see that all these different matrix proteins were represented. So, the message we got from that, and also mechanical properties, which I'm not going to show. The stiffness of these gels increased typically about 10-fold over the course of 2 weeks, partly because of the change in composition, but also partly because of the fact that the cells actually exert a tension on the matrix, and that due to the nonlinearities and the mechanical properties of the matrix actually make it stiffer. So we could actually take these matrices out with the tweezers at the end of this 2-week period, whereas initially they started out being like 100 Pascal, kind of a soup almost. So let me get back to the blood-brain barrier. So I mentioned that, we decided to go to a self-assembly approach. So we decided to take the three main cell types, endothelial cells, pericytes, and astrocytes, mix them together and see whether they would self-assemble into a three-dimensional vascular network. The endothelial cells that we used here were iPS-derived endothelial cells. They were commercially available at the time. We're now using still iPS. We're trying to go entirely over to an iPS-based system. But we're now generating all the cells ourselves. But if we take those three cell types, and this was back about 7 or 8 years ago, mix them together, put them into the region here, the gel region of the device, they would self-assemble into the structure that you see here. And the green are the vascular structures that are fully perfusible. And the other cells, the astrocytes and the pericytes, are what you see in the surrounding matrix. We can measure the permeability because we can inject a dye into these vessels, and then we can watch the dye leak. And by measuring the rate of leakage, we can determine what the permeability is, the vascular wall permeability. And we found that the measurements that we got, this is from the blood-brain barrier model, to the best that we can tell, it agrees with what you see, what you measure in vivo. You don't have measurements of permeability like this in human brains because it's just not an experiment that you would normally do. But when we compare to mouse data, it compares very favorably. We're either above or below the value at each different size or different molecular weight. So it has a realistic morphology, it has realistic function in terms of the permeability. We wanted to study it a little bit further. To look, first of all, at what's the impact of adding the different cell types to just the endothelium. So the left-hand side is just the endothelial cells by themselves. These are on the top 3 rows, those are junctional proteins. The bottom 2 are basement membrane proteins. And as you go from left to right, it's the endothelial cell culture, monoculture, to adding the pericytes, to adding the astrocytes. And what you see typically is that there's an improvement in structure, a more physiological appearing collection of vessels. The lower right-hand one is the collagen that I showed you earlier. In addition to that, we could, and this is more recent work that we've done, but with the same cells, we can now use these metabolic imaging to get deep images going through a full 500 micron depth of this tissue with cells that are completely unlabeled, label-free. This is done with the Sishin New Lab. Sishin came from University of Illinois. She worked with Steve Bogart, and she's a faculty member at MIT. And we saw that she was doing this work, so we linked up with her. And just by looking at the metabolic signature, you can identify the different cell types. So in the upper right, you can see the endothelial cells, the pericytes and the astrocytes clustered separately. So we could then look as a function of depth and identify the different numbers of cells at the different levels. And we saw, basically what we see by eye too, is that a lot of the structure was at the bottom. But as you get up after the first 100 microns, things sort of level off. We wind up getting cell ratios that are close to what you actually see in vivo between the 3 cell types that we use. I'll come back to that. We're continuing to do some work, and those are some of the results that I had mentioned that we just got last week. So when we do some EM imaging, we can compare here. Lower right is an image taken from a mouse with false coloring. There's the endothelial cell, there's a pericyte, and then the pericyte in orange, and then the astrocyte on the left-hand side. We can see the same sort of thing with our BBB model. Here you can see an astrocyte end foot interacting with the endothelium. Here we were able to capture both all 3 cell types in one image with the astrocyte end foot and the pericyte wrapping around the endothelium. So you can see that unlike the typical 2-dimensional model where you grow the endothelium on the top of like a transfer membrane, and then maybe you put astrocytes in the bottom or pericytes on the bottom, here you can actually get the realistic interactions between the different cell types. Not saying that this exactly replicates what you see in vivo, although you see later on in some of the staining that some of the images that I'll show you that we are getting pretty close to what you see in vivo. So we went one step further. I'm going to talk a little bit about the first model that we generated that included the neurons, and I'm going to step back and talk about the IPS derived model. So this is now the mixed IPS primary cell model of the BBB, and now we're adding neurons that are these Ren cells, which are, there are a whole variety of different, they're ES cells that are engineered to overexpress A beta 40 or 42 at different levels. So we can basically use these Ren cells as a model for Alzheimer's. So the top image is sort of the wild type. There's no no Alzheimer's modifications in those. The MGAP-D4, those are, it's a cell line that contains both the FAD mutation in AAPP and APP-SED1 genes. And this was published by our collaborator back in 2014. We took these and we're putting these now in a central compartment and then surrounded by the blood-brain barrier model, all of which can be perfused. And so this is what the vascular network looks like. Both in the left-hand side, you can see the control, and in the right-hand side, you can see the model that contained the Ren cells that overexpress the A beta 40 and 42. And what we started to see, and this was after 4 weeks, was that there's some accumulation. We certainly saw some accumulation of the plaques. We saw plaque formation in the interstitial space, but we especially saw a lot that accumulated around the vasculature. And we were particularly interested in that because, first of all, we could measure the permeability, and we found that the permeability was going up. So these vessels were becoming leakier. And also that this seemed to correspond with the cerebral amyloid angiopathy that tends to be one of the early hallmarks of Alzheimer's. This I threw in there just to give you an idea of the complexity of the matrix, the cells. The half circle at the bottom is the neuron cluster. The vasculature you can see around the outside, this is zooming through a Z-stack. And the sort of magenta that you see in there are the A beta plaques. So you can see it's a very complex three-dimensional model, but representative, I think, of a lot of what you see in the brains of patients. One important exception I'll be the first to point out is that our neurons are separate from our BBB, which is mandated by the fact that the two gel types, the matrix types that we use, are not compatible. The fibrin gel that we use for the vascular networks isn't compatible with neurons, so we use matrix gel for those. We're in search of a gel that's compatible with both. We've got some success, but haven't gotten there yet. But they're free to signal to one another in the sense that anything that the neurons secrete goes out into the surrounding matrix where the vasculature is, and that's why we can see the plaques forming out there. These are some particularly interesting images that we captured, where what you're looking at here is an endothelial cell in cross section, and if you look closely here, you can see that there's amyloid beta in the cell. The yellow corresponds to the colocalization of the GFP in the endothelial cells, and the red in the A beta. You can see it up in the top too, and also here you can see it's just not present in the nucleus, but it's present in the cytoplasm, which was, I think, the first observation that people have made, that this A beta early on tends to accumulate within the endothelium, and that by itself is enough to cause changes in permeability. We don't know to what extent it's important clinically, but we found it to be an interesting observation. We looked at other things, for example, pericyte coverage. We found that was reduced in the AD model. I mentioned Claudin-5 expression in the previous model, while it was even more reduced in this model. So clearly that's having an impact on the permeability of the vasculature. And measuring the permeability itself, we found that there was a small but statistically significant increase in, for example, the permeability to a 3 kilodalton dextran. We also looked at, we could sample the media both from the neuron port, but also from the vasculature side. And then we could analyze that for the amount of amyloid beta 40 or 42 that was present. And we found that in the vascular compartment, there was a significant difference between the healthy model and the AD model. So essentially what we'd expect. And even more so in the central compartment where the neurons were located, we saw huge changes, differences in the two. So at least we're getting some phenotypic characteristics of the disease. We also, and this is more recent, but again with the primary cell model, we carried the experiments out a little bit longer and then specifically looked for phosphorylated tau, because we know that that tends to show up later on in the disease process. We wanted to see whether we could capture it after 4 weeks of pill culture. And we did. We can see it clearly in the neurons compartment. We saw a little bit in the vascular compartment, but not nearly as much as the A-beta. So, clearly it's there, it's being produced. We haven't looked at the impact that it has on neuronal function yet, but that's one of the things on our list. So the A-beta that's being generated, obviously you'd like to get it cleared from the brain as much as possible. And there are two pathways it can take. And you can either go from it's being generated by the neurons. It can go to the vasculature and then go across the vascular barrier into the blood circulation and then get conducted away. Or it can go toward what are called the meningeal lymphatics. So I think I mentioned this perivascular space before. Here's the blood vessel, here's the perivascular space, here's the astrocyte feet, and this perivascular space forms like an annular tunnel that the fluid can be transported along. And there are experiments where people have introduced microbeads into the brain of mice and demonstrated, they can see the flow along these channels. They tend to disappear when you get down. They're present around the arterioles. They tend to disappear when you get to the capillaries, and then they reappear on the venous side, and then they go along the veins until you get to the penetrating veins that go across the arachnoid space up into the meninges, and then they empty out into the meninges. You can see it a little bit over on the right, and enter into the meningeal lymphatics. All the lymphatics are just beneath the surface of the skull in the brain. So we're looking to see whether we could characterize more than just the vascular compartment. And this just sort of summarizes what I was just saying. We've got this flow along the lymphatic space around the arterioles. The flow of cerebral spinal fluid goes through the parenchyma, enters into the glymphatic space on the venous side, and then goes up into the subarachnoid space, into the dura, and then into these meningeal lymphatics. That's under normal conditions, and in Alzheimer's, what happens is you get diminished drainage of the CSF. We know that a lot of this flow occurs during night while we're asleep, and it tends to be impaired in cases where you have accumulation of A-beta in the brain. So the model that we developed for this, and I guess you'd call this more of an engineered model. We took the devices up here. You can see the schematic over on the left. The pink region is where we would introduce the blood-brain barrier cells. The yellow region is where we'd introduce the neurons. And then we had a separate gel compartment on the top there. This is sort of looking down on top of it. And then on top of that, we would introduce the lymphatic cells. And the lymphatic cells we could induce to sprout into the gel by means of introducing an interstitial flow in this diagram, from bottom to top. Okay? So as we introduce the flow, these vessels sprout, and then they become functional lymphatics. This is what the system looks like when it's all complete. The neural compartment is at the bottom. You can see the green BBB, and then the lymphatic vessels are also on the top. And then there's the box that just shows a blow up of the lymphatics there on the left, and then a blow up of the BBB where we flowed in a dextran dye. So you can see that these are all fully perfusible vessels. One interesting result, and this is preliminary data, and obviously we need to make sure, we need to confirm it, but what we saw is that when we developed the lymphatics in the normal situation with no A-beta present, the lymphatics form very aggressively. If we added A-beta to the system, both in 6 days after seeding, saw that the growth of the lymphatics was very much stunted. by the presence of the A beta. There are a number of factors that could contribute to this, but we feel that this might be an important factor in terms of limiting the outflow of the amyloid. So we, at that point, had a kind of a mixed model where we had the iPS-derived endothelial cells, and we had primary cells from different patients, the astrocytes and the pericytes. And for any of you that have done experiments with these systems, you know that anytime you change one cell type, which you have to do with primary cells periodically, you have to re-optimize the system. So what we did is we wanted to see whether we could develop a purely iPS-based system so that we'd have a consistent source of cells over long periods of time. So again, we're going to do both the blood-brain barrier and also the neurovascular models with the AD or PD neurons. And this is kind of, that's the device that we use. The center port is where the neurons are put down here, and then we put the BBB on the outside. So I'm just going to step through and talk about the different cell types where we got them. The endothelial cells, there was a recently published differentiation protocol where you use transient ETV induction. It turns out it's a beautiful method for generating endothelial cells in a relatively short period of time. You can see that it's only a 5-day protocol, and then you expand the cells afterward. And it's a very highly pure, it's a very pure population. If you look at the marker CD31 or VEGFR2, receptor 2, you can see that a large percentage of the cells express both of these markers. They also proliferate very rapidly. This is the protocol that we're using. This was the previous protocol where they didn't do a transient induction, but they just used a continuous induction of the ETV2. And this is the networks, these are the kinds of networks that we grow. And this is a typical picture. So the vascular structures, you know, they're nicely formed. And if you compare this side by side with the brain capillary bed, you see that there was, in terms of morphology, it's very similar. This is a 100 micron scale bar, and you can see the vessels we're getting range from, you know, probably about 10 microns up to 20, maybe 40. So it's the dimensions of the type of size that we would like. We did some bulk RNA-seq just to see whether the right genes were being expressed. And what we did is we compared our iPS-derived cells, the iPS-derived ETV2 cells, to human brain microvascular endothelial cells. So those are primary cells. We did this largely because, well, we wanted to characterize them, but we also wanted to check to see whether our iPS-derived cells had the same problem that the previously published brain microvascular cell had, where it turned out that they expressed a lot of epithelial markers. And this was a study that was done after the initial work that was published, and showing all the differences between, in terms of surface markers, the Claudin family proteins and barrier properties between the primary cells and the iPS-derived cells. So we're specifically interested in things like the junctional proteins, which you can see here. I can't see it very well here, but Claudin 5 and also PCAM1. Also a number of a lot of the RNA over here, the genes are those that would be highly expressed in epithelium and not brain endothelium. So we found that there's some that we do get highly expressed in, in the iPS-derived cells. Some of the ones at the top, but the primary ones we're looking at, Claudin 4, Claudin 6, Claudin 7, which are primarily epithelial, we get very low levels of expression. So, and this is all in 2D before we co-culture these endothelial cells with the brain cells. So we think it's going to be even better. Those are the experiments that I had mentioned where we're just collecting the RNA right now. We also need the astrocytes and pericytes from the same iPS source. We're using this Alstem 11 cell source because it's readily available, and also the companies that we're working with can use it for commercial purposes. So we use a protocol that was developed by our collaborators, Rudolf Janisch and Maya, Mitoflova, from deriving the pericytes from neural crest cells. And there's another protocol, published protocol by Perio et al. For deriving the astrocytes. The astrocyte protocol is quite long. The pericyte protocol is not as long, but it's a little finicky. But fortunately, we have their help to do that for us. So we get those cells from them. We derive the endothelial cells ourselves. The other cell type that's probably critical are the microglia, which I haven't talked about at all. But if we want to have an immune competent system, the microglia are turning out to be a really important cell type in the brain. So here you can derive the microglia from iPS cells. These are actually derived, I think, from the Alzheimer 1 cells. And just to demonstrate that they can be grown, they can be differentiated, they have all the morphology, and they've analyzed the markers and also the expression patterns of these cells. So they seem to be true brain microglia. And we plan to add those then to our system. Not yet, but at some date in the future. The BBB itself, this is a collapsed Z-stack on the left-hand side, and then on the right, you can see we're sort of stepping through the Z-stacks. Again, this is showing GFAP, which is the marker for astrocytes, PDGFR, which is the marker for pericytes, and then the UEA, which is the marker for the endothelial cells. So, and here you can see that there does seem to be good wrapping of the astrocyte end feet around the endothelial cells. And also for the pericytes, the sort of the pinkish color. So we tend to get good organization, and the cell ratios that you see here are pretty close to what we see in vivo. And again, we went back to Sishin's lab and looked at the distribution of cells within our iPS-derived system. And it turned out to be very similar to what we were seeing before in terms of, again, a higher cell density at the bottom. But as we got up into the bulk of the system, the cell ratios were pretty close to what you see in vivo. So we were very happy with that. We also looked at then the permeability of the systems. And I'll show you some data on that, which also agree with what you'd see in vivo. So what we're looking at now is a completely iPS-derived model for the blood-brain barrier, including the astrocytes, the pericytes, the endothelial cells. We were also interested in some of the transporter proteins, and in particular, these three, RAGE, LRP1, and then, well, 4, PGP and BCRP, because these are all instrumental in terms of transporting amyloid beta across the endothelium. We started by, first of all, looking at just sort of a panel of molecules of interest, the dextrans, because we always use dextrans to measure permeability, transferrin, because the transferrin receptor is being used for shuttling a lot of drugs across the endothelium, and then a few others that we've started to test. And these are some of the results that we've obtained. This would be for permeability of different dextrans. And the way we do this, as I think I mentioned, we load the vasculature with a fluorescent dye, and then we observe the leakage from that vasculature over time. From that, we can calculate what the permeability is. And you can see we get a very nice size-dependent permeability distribution for the different size dextrans from 3 kilodaltons up to 150 kilodaltons. 150 is relevant because that's about the size of an IgG. We could also look at the permeability. We're interested not just in the permeability, the leakage from the vessel out into the surrounding tissue, but also from the tissue back into the vasculature. In particular, things such as amyloid beta, what's the permeability of that fluid back in? So first of all, we thought we'd try looking at some of the dextrans because we'd expect that to be symmetric. We'd expect to get the same permeability one direction as the other. And here I just got the data here for one size, a 40 kilodalton dextran, but we found that we could indeed get the measurement techniques are such that we get the same permeability, which is reassuring. Sheng Yu has now done this for 5 different sized dextrans, and they all agree in terms of the direction of permeability. So that enables us now to look at directional transport with these different transporter proteins that we're interested in. So getting back to the transporters, here is images of PGP on the left, BCRP, and LRP1. They're all shown in red in these images, different experiments. But you can see that they're all localizing to the vasculature, and they all seem to be present in our model. So we know they're there. And so the question is, are they functional or not? One thing, not only are they there, but they're also localized in the right spot. So for example, with the PGP, we'd expect that to be on the luminal side only, not on the abluminal side. And we can see now from some of the images like this one, here's the nucleus of an endothelial cell. This is the abluminal side, the luminal side. You can see there's a faint red there on the luminal side of the nucleus. So it's very clear that these proteins are being at least predominantly expressed on the luminal side. And we could see the same thing for, here we're looking at PGP, BCRP. And again, we see the same sort of thing where, again, here's the image of a nucleus. This is the lumen over here. You can see the red staining just inside the nucleus. And the same thing up there for PGP. So they're present, they're localized at the right spot. Are they functional? So here we looked at adding mannitol to the system, where mannitol acts on the blood-brain barrier by basically shrinking the endothelial cell some. So it opens up the gap between the endothelial cells and therefore increases the permeability. So first of all, we demonstrated that when we add mannitol, the permeability for both 40 and 70 kilodalton dextrans go up. We could also look at the transferrin receptor. And here, we're looking at the linkage from inside of the vessel to the outside. Transferrin being actively transported via the transferrin receptor, we'd expect the permeability to be higher. But if we inhibit that receptor, we expect the permeability to come down. And in either case, it should be higher than a similar molecular weight dextran. Okay. And here what we've done is we've taken IgG and then an IgG that's functionalized with the transferrin receptor and shown that we get a significant increase in permeability. Again, everything seems to be checking out with what we'd expect. And this is it's a movie that we just got last week. What we're looking to see now is if we could capture the leakage of a drug from the vasculature into the surrounding tissue and the uptake by the different cell types, the endothelial cells, the pericytes, and astrocytes. So we saw from before, these are all unlabeled cells. What we did is we flowed in a fluorescent transferrin, and we then observed that leakage of the transferrin into the surrounding tissues and the uptake by the resident cells, both the endothelial cells, pericytes, and the astrocytes. And to the extent that we can identify and distinguish the different cell types in the sort of gemisch here, and it gets very difficult to do that. We're able to track the increase in absorption by the endothelial cells over time. The way they do that is to take the initial image, and then from every subsequent image, they subtract the first image. So the dye in the vessel disappears. Okay, so we're only looking at the dye that leaks out in the time from that first image. Okay, so here, show the movie again, but basically you can see over time, and this is about 1 hour, that essentially the dye permeates and gets taken up by the cells. And this shows essentially that these are primarily endothelial cells that are uptaking the transferrin. Again, which is what we'd expect, but to my knowledge, it's the first time that we've actually been able to observe drug uptake in a transient experiment in real time, even in an in vitro setting. So we taking the BBB model, it's iPS cell based. We're now putting back the neurons to make our, I'm sorry, here we're still looking at just the permeability, and here we've done is we've looked at the effect of the A beta on permeability, looking at 2 different sized X strands, adding exogenous A beta to the system, and then looking at the change in permeability. One of the problems that we had here is that we saw that as we went to very high concentrations, the system, the vascular system starts to deteriorate. Okay, so basically, part of this increase in permeability is the fact that probably some of the vessels being partially destroyed. Here we're adding the organoids that are derived from the iPS-derived cells, and these can be either Alzheimer's or Parkinson's. And what we do is we form those organoids, we can place them into that central channel rather than seeding the cells separately. We just put the organoids in. And then we can look at the co-culture over a period of time to see whether, in fact, we get the same type of phenotype. So these, again, have the point mutations for AD and also for BD. And I see that I'm running a little over time, so let me finish up here quickly. Just to be short and sweet about this, we start to see some effects, phenotypic characteristics of the disease process in as little as 2 weeks, 14 days. We see an increase in permeability both in the Alzheimer's model and also in the PD model, increases by about a factor of 2. And so that's where we are right now. And some of the things that we're doing, in the process of doing, we plan to establish a collection of patient-specific models that we can use then to get more, rather than just using one iPS cell source, we want to be able to get these from multiple, both healthy patients and also Alzheimer's patients. We're looking to add the immune system, both the adaptive and the innate immune system, primarily in terms of the innate immune system, mostly the microglia and the macrophages. We have systems for adding continuous flow. We just need to connect them up to these systems, which adds a little bit to complexity. And then ultimately do use computational modeling to sort of pull everything together, the leakage from the vasculature and transport out through meningeal lymphatics. So with that, I'll end. I just really have to acknowledge a lot of hard work and a lot of really good students and postdocs that have contributed to the work, primarily the ones that are shown in blue here, and the funding sources that we have. So with that, I'll stop and be happy to answer any questions that you might have if there's time.
Questions?
Hi, thank you for this talk. This is all really fascinating. So I have a question about your neurovascular model of neurons. So I've heard that the diversity of neuronal populations can vary depending on the region of the brain, and I was wondering if you expect your model to be more applicable to some regions.
So I wouldn't say the REN cells are specific to a particular brain region. We are, when we develop the organoid systems, those we are generating, there are different protocols to develop, for example, cortical neurons or, midbrain neurons. We're trying to match the region of brain we differentiate those organoids into to the disease model, where the disease occurs. But with the REN cells, probably.
Amazing talk, great work. I was just wondering, are you able to control the pressure during perfusion? And if so, does that have any lasting effects on during the duration of perfusion?
So perfusion by itself has a dramatic effect on the architecture of the microvasculature. We haven't done it in the BBB model yet, but we've done it in sort of our generic model with cuvex and fibroblasts. The vessels remodel considerably. The vessels that are conveying most of the flow tend to widen. The ones that are sort of perpendicular to the pressure gradient tend to regress and disappear. We have found a way to stabilize the system. Basically, what we do is after 1 week, after a couple of days of flow, we take away the growth factors from and just use maintenance week, and that seems to work. So we have some control over the architecture and the morphology, but not. Oh, it definitely is, yeah. Yeah, we've done some other studies where we've looked at interaction between the cancer cells and the glycophilics on the endodermal. It's definitely there.
Other questions?
Do you think the morphological response to pulsatile flow is different to the response to steady flow?
Well, the pump that we have actually has about a 10% pulsatility, and we do it at 1 Hz frequency. And since we're talking about the microcirculation, that's probably not too far from what you actually have. Whether the pulsatility has any impact, I can't really say. I mean, the other flow system that we use is just a hydrostatic head. So that's continuous flow, but it's an intermittent flow. So the two really aren't directly comparable. You know, the other systems that people have, like there's a Nemetos platform that just rocks back and forth. That basically it makes flow in the media channels, but doesn't generate a pressure gradient across the gel region. And so there really aren't many good systems that we can look to where we can answer that question. I think with our pump system, basically there's a capacitor that we can change the value of that will affect the pulsatility. So we could, in principle, look at that question that we have.
Other questions? Sir?
Great talk. That's a main question. Can the Vitro model system be used to study the effects of mechanical stress or agitation on the accumulation of A beta, for instance, with the mechanical stress in trauma, or therapeutically might be used for influencing lymphatic drainage or conversely increase the accumulation of A beta due to some other.
Yeah, absolutely. I don't see any reason why it couldn't be. We've talked a little bit with Peter Long at UCLA. You know, he has these ultrasound methods that he uses, so we can do some mechanical stimulation that way. We haven't tried to do that in our microfluidic system very much, but we're very much interested in the role of mechanics. We're doing some experiments to look at the mechanical properties of the cells themselves and how that changes with AD. So, for example, looking with in Glow, you can introduce small beads into the cells and then use the oscillation technique or just drag it to infer the real and imaginary parts of the modulus. So, and we're also looking at things like piezo-1, the mechanosensitive protein, because that seems to be implicated in some aspects of Alzheimer's, and we find that it's expressed in all the cells that we're working with right now. So it could have important implications.
Great. Well, let's thank Professor Kamm for really good.